Okay, so today we're going to go over the answers to the FRQs you did this week, and the first one is about yeast cells and their survival ability in different environments. So if you need to pause the video now and reread the question to familiarize yourself with it, I recommend that. But if you're ready to go, the first thing I'd like to say is that this is a very common experimental strategy, right? So one way to study a gene's function in an organism is to intentionally mutate that gene and then see how the organism survives with that mutation. And you can conclude then, as a scientist, that if that organism doesn't survive with the mutation, that mutation is linked to the situation you're putting that organism in. So, or rather that the function of the gene that was mutated is linked to the growth medium. So part A says identify the control of treatment, identify the role of treatment 1 in the experiment. I think that the correct answer is treatment 1 is the control, period. I don't think you need to elaborate because part A says identify, which means point it out. However, I was watching a video where there was an AP, like College Board employee describing the answer, and she said she wanted to see treatment 1 is the control because it tests the viability or survivability of the yeast, meaning it tells us that all the yeast cells were living and that they're not dead because of another reason. And it attributes the results of this experiment to the presence or absence of amino acids and not some other factor. Excuse me. I think this is nice. I think if they wanted that, they should have put identify the role of treatment 1 in the experiment and describe how it serves that role. She may have just been covering her bases, but there's two ways, I guess, to answer that question. One is just to say it's the control, and then the other is just to elaborate on that. For part B, it says provide reasoning to explain how mutant 1 can grow on treatment 1, but not on treatment 3. And here I have my explanation, which would be given full credit. Methionine is necessary for yeast survival, and that's present in treatment 1, but not treatment 3. Therefore, the yeast cells in the experimental group called mutant 1 must not be able to produce methionine. I think that's pretty self-explanatory. The third part, part C, is not. So the third part says, these are haploid cultures, and when they mate, they make a diploid cell. And if a researcher mates mutant 1 and mutant 2 to produce diploid cells, can we predict whether or not those diploid cells will grow in each of the 4 media? Excuse me. So this is where we would write our answers, and I think this one's pretty easy. If we mate this haploid cell with this haploid cell, and they both were able to survive when all amino acids were present, they're going to continue to survive. This one, treatment 2, I think is strange. The College Board says that this should be a positive, and I say it should be a negative. And I'll come back and describe that later. These two, if we mate a cell that can make leucine when it's not in its environment, and we mate a cell that can make methionine when it's lacking in its environment, and if those are haploid genomes, meaning they have a half set of chromosomes, then when they combine, they will be able to survive, kind of like having the best of both worlds. So the diploid organism would have the benefit of both this gene or ability to produce methionine and this one, the ability to produce leucine. The College Board says that, excuse me, this should be positive, meaning that the yeast cells, the diploid cells produced by mating, would still be viable. I disagree with that. I think if there's no amino acids present, that they won't survive. And the only 2 amino acids they tested lacking in the environment were methionine and leucine. And you know there's 20 amino acids. So I would say the answer is no. They're arguing that the diploid cells would survive because if you take a yeast cell that doesn't require methionine, and you take a yeast cell that doesn't require leucine, and you mate them together, the fact that these guys died over here and these guys died over here is erased because, again, we're combining the two genomes to make a diploid genome. I don't think that explains the other 18 amino acids, though. And this says no. So I've watched a couple videos on YouTube about this, of other teachers explaining it. They say that the answer could go both ways. I know that's not satisfying for you, but that's kind of part of the test, using your noodle, using your noggin. All right, I'm going to stop this video and start with the next question.